Okay, I want to welcome you one more time to EHE Community Connections. We want to remind everyone viewing that this meeting that the EHE Foundation does not provide medical advice. The information provided here for you today is just for your information. We encourage you to take all of that information and any questions to your healthcare provider. And now I turn the mic over to Denise to begin today's discussion.
Hi, everybody. Good morning. Good afternoon. Great to see you. I actually can't see all of you. I'm so glad that so many of you have joined today. As Julie said, my name is Denise Robinson. I am the Director of Research for the EHE Foundation, and I'm also on the Foundation Board. I'm here today with Hugh Leonard, who is Chair of Trustees from the EHE Rare Cancer Charity UK. Thanks for being here, Hugh. And we are excited to be talking with you today on Saturday about science. We realize that you probably had some more fun things that you could be doing today, so thanks for joining us here today. We're excited to give you an update. This is meant to be a high-level overview so that you will know what's going on in this community where excellent research is happening around the world. And before we start, the first thing I want to do is thank patients, EHE patients, for contributing to this community. Researchers, I think that there are going to be a couple on the call today, so thank you for giving parts of your lives to us, and also to clinician scientists who are working to fight this disease and find a cure. The commitment of this community has made today's progress really possible, because without all of you here, we wouldn't have anything to report. So let's jump in. I see that people are still being admitted to the room, so welcome to all of you who are joining us a little bit late. So just to get focused on what we're going to talk about today. So this is meant to be a high-level overview. Again, I'm going to give apologies and shout outs to all of the researchers who are here and who are not here. We are not going to be able to appropriately go into all of your research, but we're going to give a good overview. But why are we here? We want to focus on that. We want to remind everybody that patients and advocates are essential to rare cancer research. Rare cancer research is underfunded, understudied, and you're essential because nothing's going to happen without you. And then the next thing that we're going to dive into are progress and current initiatives in EHE research around the world. And then the big thing, why are we here? Our mission is to find a cure for EHE. We work towards that every day. And to find effective treatments or a way to stabilize the disease so that people can live a good life with EHE. Now I'll turn it over to Hugh.
Thank you, Denise. Hello, everyone. It's fantastic to be with you all here today and have the chance to provide this overview. I'm going to crack straight on with this because we've got a lot of ground to cover. And what we wanted to do in the first couple of slides is just emphasize this issue of patient participation and advocacy. And what we wanted to start with was just this simple diagram that hopefully you're all looking at, which I call the cancer collaboration. And around this diagram, you can see the main players, the patients, doctors, government, pharma, researchers, funders. This model is very powerful for common cancers because of the sheer numbers of people involved. So basically, it's self-driving and self-supporting for common cancers. If we look at this model, however, from the perspective of an ultra-rare cancer like EHE, it's actually a very different story. Patient numbers are tiny. The number of doctors involved with them is also tiny. So their collective voice is very small. Government has lost interest now, really, because there's no political driver. There's no huge group wanting to support the government or government wanting to have their support. Costs to the national budget of treating very rare cancers is very small. Pharma's not interested because they don't see any particularly strong profit potential in rare cancers. Researchers tend to be turned off because it's just not seen as sexy and there's no data. They see limited data, limited samples, etc. And without the clamor, funders may not even know we exist. So faced with that, people with rare diseases have really one of two choices. We can either feel sorry for ourselves and just hope that fate is kind to us, or you have to organize and mobilize the patient community, and you have to get into the center of this circle, and you have to drive the process. We, the advocates and the patient community, need to make sure that our voice is heard by governments, and we need to drive pharma, if through nothing else embarrassments, to invest in the research. We need to go and tell researchers that we're here, we have samples, we have information, we have data, and we need to go and find the funders and tell them that we want them to fund rare cancer research. So this really comes down to showing that we have people who are going to participate, showing that we can provide numbers and data and information, etc., and also doing the fundraising ourselves, because we cannot expect people to give us money if we're not doing it ourselves. And if we do all of that together, and we are doing that together, we create real momentum, and it's the momentum that drives the process. So the question, I guess, is, has it worked for us? And I think if we look at the research position, this is what 2015 looked like, EHE research. Dr. Brian Rubin was, as far as we understood, the only person who had a dedicated, very small team at the Cleveland Clinic. If we now look at the 22 position, 2022, we see a very different story. Don't try and read what's in these boxes. They're all tiny, and we're going to come on and talk about most of these a bit later on. This is just for visual impact, to show you the difference that you, the EHE patient and supporter community, have made. It's your money, your fundraising, your samples, etc., that have allowed us to drive research to a point where on this chart, there are 25 projects, research projects going on that we are involved with around the world. We're not funding all of them, but in most cases, if we're not funding, we're directing EHE patients to them to provide data and information. We've got also, this is not a complete, this was a slide we put together in middle of 2022. And since then, we've added 3 or 4 additional projects. The EHE Foundation have funded 3 new projects this year, and those grants have gone out. And we've had a huge donation, a fantastic donation in Canada that's allowed a really substantial piece of research to start in Canada. And we're going to talk about that later. So that is fantastic. If we move on to another example of how momentum helps us, in 2021, Silvia Stacchiotti chaired the ESMO consensus paper process in Europe, but involving many people from around the world, specialists, and I've highlighted in yellow some of the names we'll recognize, to produce the first ever consensus paper where everyone came together, all the sarcoma specialists of all the different disciplines, and pulled together and agreed what was the total picture of EHE in terms of treatment and what needed to be done. This was fantastic, and we were able to then produce the patient version that some of you will have seen. This, I don't believe, would have happened if we hadn't had real momentum behind the EHE process that our community has driven. So that's all I'm going to say, just to drive home, this is why participation is so important. And you will see, and I'm sure some of you have thought, foundations are banging on about samples and Biobank again, or they're banging on about registry. This is why. It's about momentum. It's about proving to the people out there who can help us that they should engage with us and help us. Okay, enough that's from me. Back to you, Denise.
So when we as advocates, and I'm going to talk about the EHE group here, primarily the Australian EHE Foundation, the UK Rare Charity, and the EHE Foundation, we sit down and we talk together about what research is going on. We look at prioritization of projects for funding, the significance of those projects, what are they going to tell us? And we have started grouping things into 2 buckets. So we have research enabling projects and basic science and translational research. And we want to talk about those in that way today. Research enabling is basically what you think of, right? It's creating data that's needed in order to do something else. It's foundational. And then the basic and translational research is understanding the disease better, leveraging that enabled data that we've gained, and investigating therapeutic targets so that those can be researched in the clinic. So we'll jump into the enabling first. So data and biological samples. At the early, and I don't know if you can see my cursor on my screen or not, we have to have patients. So we have a disease in a patient population. We have EHE patients. These patients need to be connected to researchers. There are researchers who are interested in studying that disease. And by those patients all coming together, you form enough mass of people, a group of people that researchers say, aha, there's something to study here, and there's an interested group of people in the diagram that Hugh illustrated. And then patients, people with the disease, become participants in research. They start giving to that research, making it possible, enabling it essentially. And then you have those ways that people are enabling research using, for instance, a centralized biobank. Already today, we have excellent enablers in progress. We have the US EHE Biobank at the Cleveland Clinic that is collecting specimens. We have the UK EHE Biobank at the Royal Marsden. The Australians have an ongoing biobank. And then there's some data studies that are going on. The NCI MyPart, you all have seen that promoted in our community as a rare tumor study that's funded in the US. The ISCIS study in Australia is another very important study that's generating data. There's Cartwheel, also out of Australia, that's generating data. And an important study that was done a couple of years ago that detailed quality of life of EHE patients. So these are all projects that are in progress. They are live, active, and generating data for this community that are going to power more research to happen. And then if you look over on the right side here, there are initiatives that are in progress. So these are things that are not yet live. They're not ready to enroll participants. But I think what you're going to see in 2023 is a real explosion. And be ready, because we'll have a lot of information coming out about these. But these are all very important. So you'll have a pan-European study that's coming out across Europe. And some of these are different. So you have different ways that data and biological samples are captured and harnessed. Some of them are patient-donated data, so patients are asked to tell their own story, maybe to answer surveys, participate in a registry where you tell your journey. Sometimes that data is captured through a clinician's perspective or through medical records. So if you look at like the Pan-European Perspective Study, that will be a medical-focused study. The patient registry will be patient-entered data. We have an ongoing up on repurposing sirolimus for EHE, and then the ProCare EHE Canadian research. So these are all going to be large initiatives that you'll see. They're going to enable much more research to happen. They will, by themselves, generate data. They'll tell us something about EHE, but also the data that's generated is meant to spark other research that's going to happen. And I want to give you just a snapshot. This is a end of year from 2022 cut of the data for the US EHE Biobank. And this is just a proving ground here. We've made great progress. EHE patients are contributing meaningfully to research. We have now 60 samples, or I'm sorry, 60 participants engaged, not 60 samples collected, but over 60 participants engaged. And already we've started this year with more people being engaged in the biobank. This is, as all of these biobanks are, this is a centralized way to collect ultra rare tissue that is very, very precious in order to drive other research model generation. And in the US perspective, particularly with the guidance of Dr. Rubin, we have the benefit of every sample being evaluated, validated as EHE. And then our goal is not to keep these specimens in a bank. We're not just growing these here. The goal is to move these out for research, because when we can get them in our bank, validated, and then out of our bank, then we can start other research like model development. So it's really, really important. You see us, promoting this all the time, but I can't tell you how precious it is. Anytime there's a surgery, a fluid drain, it is very, very important that we can get those EHE tissues and fluids into our biobank. And here really, this is tissue collection in real life, working in research. I think this is probably one of the proudest things that the EHE community overall we should be proud of. We have a genetically engineered mouse model. Dr. Rubin's lab created this. It is already producing data. So this is Dr. Rubin's gem is a close analog to human EHE. The advantages of this model, it has an intact immune system. So you can, and I think I see that Dr. Rubin is coming into the call now, so hopefully I'll get this right. You can study the immune system of EHE. You can study EHE in an intact immune system. And then complementary to that, we have a PDX that has been developed at the INT in Milan. A little bit different. These are in compromised mice, so they don't have an immune system. But both of these models are very, very important for better understanding of the disease and for drug screening. Super important. Dr. Kouskoff's lab is going to be working with stem cells, again, to screen a classification of drugs called PARP inhibitors. And then we have other models that are in development. Something to know is that human trials can't take place without preclinical work that's been done in these models. And just because we have these model systems, that isn't enough. We need more models. We need more models of the same types of models, and then we need different models. Different models look at different things, look at disease progression, disease biology, and again, drug screening. So very, very important for this community and great progress on this too. Hugh, I'm going to take it back to you.
Yeah, thanks, Denise. We're going to go on and talk now about some of the other biological research and translational research, but we thought it might be useful to just quickly, because there may be people on this call who are quite new to EHE and haven't seen this sort of thing before, just to remind people about some of the biology of the disease. And this diagram is a genetic example of a cancer driven by a chromosomal rearrangement. On the left-hand side of this diagram with the gray background is the normal cell. And on the right-hand side with the pink background is the cancer arrangement or cell. And if we look on the left-hand side, we see we have a pink gene and a blue gene. They create, those genes are pure, they're pink and blue, and they create proteins that are pink and blue. And that's what you should have in your cells. And those cells will then go ahead and control cell proliferation and regeneration. At the top, however, we can see what happens when we have a translocation or a rearrangement of the chromosomes. Those two genes, gene A and gene B, they break, and the two parts then cross over and reconnect. And so now we have an arrangement of the genes that shouldn't be in our bodies. And so rather than having a pink gene and a pink and a blue gene like we see on the left-hand side, we now have a fusion gene that is a combination of these things. It's pink and blue. And it creates a fusion protein that is pink and blue in this diagram. And that protein shouldn't be in our bodies. It shouldn't be there, but it is there. And it's in our cells, and it thinks, hey, I'm here, I may as well start trying to do stuff. And so it off it goes into the cells, and it starts creating a little bit of chaos. And in the case of EHE, of course, it creates a lot of chaos because ultimately what we face is a cancer. So what we know what the chromosomal rearrangement is in EHE. Dr. Rubin and others found that in 2011. We know the fusion proteins that we are dealing with, TASCAMTA1 and YAP-TFE3. And we're beginning to understand the actual biology of how those fusion proteins are actually affecting ourselves. And what we really focus on now, what we're focused on with our research is the bottom of this diagram, which is the actual process of cell growth and regeneration, and how are these fusion proteins affecting us. Now, there's a couple of tiny bits of terminology I just want to mention because they may be useful later in the slides and if you go on. Hippo pathway. The Hippo pathway is the name given to a very complex process in our cells that controls cell regeneration. So when your body needs new cells, it's the Hippo pathway that makes sure you get the right cells in the right place, in the right number. We know that the fusion protein is impacting that Hippo pathway in EHE and corrupting it. And that's why we get a proliferation of cells and tumor growth. The other thing I just wanted to mention was is around the generation of proteins. Protein generation involves two steps, transcription and translation. And it starts when a protein binds to our DNA. It comes along and it attaches itself to the DNA at a certain point, one end of the protein. The other end of the protein's waving out there in the cell looking for something. And another protein comes along and goes, hey, I'll hold your hand. So we have two proteins connected. And so that may go on for a little bit longer, and you may have a few more proteins. And those proteins will ultimately go through a complex process that I'm not going to go into, but that will create more proteins. The point is that TASCAMTA1 and YAP-TF3 cannot bind to our DNA. So if we think of the DNA, the protein that attaches to our DNA as protein 1, and the protein that attaches to that as protein 2, these fusion proteins can't be protein 1, but they can be protein 2 if they can find a protein 1 to attach to. And that is what happens in EHE. And the protein that binds to DNA in EHE is TEAD. Now, I think there are 4 TEADs, in fact, and it's TEAD4 that is the protein that starts the process for EHE. And then the TASCAMTA1 or the YAP-TFE fusion, it comes along as protein 2 and it attaches to TEAD. That is fundamental to the progress of the disease. If we could stop TASCAMTA1 or YAP-TF3 binding to TEAD in our wildest dreams, we could kill this cancer or have a huge effect on it. And so when you see reference to TEAD inhibition, that's what we're talking about. We're talking about inhibiting the ability of TEAD to bind with TASCAMTA1 and TF3. Transcription is the overall process we see. They're terms you'll see in the later slides, so I just thought they were worth just mentioning here. Back to you, Denise.
Okay, Science Saturday. I love listening to Hugh's enthusiasm in that conversation. That's great. So we're gonna shift focus a little bit, and the next few slides we're gonna go through, we're gonna talk about some labs. Particularly, I'm starting here with Dr. Rubin's lab, and then we'll go into other labs and country initiatives. Again, I'm gonna make an apology and a statement here that we are not going to even do remote good justice to the science that's going on in these labs. But we wanted to make sure that you do know that there is great work going on around the world, and highlight some of these places where it's going on. I mentioned earlier, I think that Dr. Rubin joined, but I can't see all the participants because of the view I'm in. So I don't know, is Dr. Rubin on and in any capacity to say hello or speak specifically to research going on in the Rubin lab?
Yeah, I'm here.
Hey, Dr. Rubin.
Hey, everybody. Sorry, I was at the Saturdays in my household are just running from one sporting event to the next to watch my kids, but we're in between basketball, gymnastics, and soccer right now. Just came from a basketball game. Anyway, yeah, Denise, what do you want me to run through kind of what we've been doing in the lab? And
Yeah, sure. Talk about the highlights, features, new things, anything you want to, just for a couple of minutes. We won't keep you long.
Okay, well, I mean, yeah, we discovered the gene fusion back in 2011, and it was essentially another laboratory, that of Christina Antoniescu at Sloan Kettering, simultaneously discovered it, and they used a different technology than we did. And so it was nice kind of instantly confirming 2 papers published literally at the same time describing this gene fusion. And, early on, we recognized that there were really no models for this disease. There were a few people that claimed to have models, but they really didn't. And so we put a lot of effort into developing model systems. And the reason that you want to develop model systems, if you have any interest in studying biology, you need relevant models. And so we started to make a genetically engineered mouse model, and we started to try to culture human EHE cells. The culturing of human EHE cells has been, I don't want to call it an exercise in futility, but we have never been able to develop a EHE cell line from humans for whatever reason. Since we can't do it, we really don't understand even why it's not happening. However, we did get the genetically engineered mouse model to work, and we published the first paper on that in 2021. And it's a really nice model in that we basically just make the same genetics that are found in human EHE, but in a mouse. And so those mice develop only EHE. They look and act and are genetically identical to the human disease. We then actually went and engineered version 2 of the mouse, where Hugh and Denise did a nice job of describing the background that EHE is a simple cancer. It has typically one genetic event in it, which is this gene fusion. But in about half the cases, and these are typically very aggressive cases, there's loss of one other gene, and it's what's known as a tumor suppressor. There's a bunch of tumor suppressors. I know you're not scientists, but you may have heard of something called P53. P53 would be a typical and canonical tumor suppressor. We actually removed another tumor suppressor known as CDKN2A, which is also a typical tumor suppressor, and it's lost more often in EHE. So we actually combined 2 mouse models, one that had the EHE gene, and then the other one that had loss of CDKN2A, to make a newer model. And lo and behold, we do get more aggressive tumors. And the nice thing is that we're able to develop cell lines from those tumors. And so since they've lost CDKN2A, it really allows them to grow in plastic. And so we have developed a bunch more models from the genetically engineered mouse model. And it really allows us to do a lot of studies. So we actually have gotten 2 grants recently. I haven't announced this one yet, but we got a grant from the DoD a couple of years ago. Actually, I think this is the last year of that grant. But we were just awarded a grant from the National Cancer Institute NEXT program, which is the experimental therapeutics group there. And it's coming out of something called the Chemical Biology Consortium. Essentially, they're going to give us money and resources that allow us to develop our own anti-EHE drug. And as Hugh discovered or described, we are trying for something called a PPI, which is a protein-protein interaction inhibitor. And it's we're trying to inhibit that interaction between TAS-CAMTA1, the gene fusion, and TEAD. And think of TEAD as an obligate partner. TAS-CAMTA1 just doesn't work without its ability to bind TEAD, at least in the things that we're interested in. So we just got that grant. Actually, we're going to get the letter of award pretty soon, but we went and got the award right before Christmas. We don't have a chemical group that we work with, a bunch of chemists to develop these drugs. We have a YAP1 TFE3 gem that we've developed, and we're just starting to study it, but we have the mice now in our facility, and we are just starting to try to get them to make tumors. I'm not going to describe that a lot more, but YAP1 TFE3 is another thing that we're kind of interested in, and we actually collaborated with a patient group who dedicated some philanthropic funds actually to help us make that model. And we actually have the model in-house now. We've got a lot of drug screening going on, trying to identify drugs that are FDA approved for other purposes, to see if we can identify other inhibitors that don't exploit this protein-protein interaction. And as is listed here, we did launch the phase 1 clinical trial with Novartis to study their TEAD inhibitor, which is known as IAG 933. And that trial is actively accruing. And I don't know much about results of the trial. A phase 1 trial is really designed to look at toxicity of the drug. And so they do dose escalation. They start with a small dose. If the patients are doing okay, they will then start increasing the dose. And that is where that trial is currently. And I would say there's 2 other clinical trials. So again, we have a litany of other studies that we're doing in the laboratory currently. We're interested in the microenvironment and how cells that surround the tumor. I mean, cancer cells are not just pure cancer cells. They have lots of other cells in them. And we're interested in what the other cells that kind of bind to the cancer cells and interact with them are doing. The idea there is that if they're doing something important, we might be able to drug the other cells as well. I would say that we just had a paper that just came out in a journal called Clinical Cancer Research which describes in detail volume 2 or version 2 of our genetically engineered mouse. So if people are really interested in the details, you can read that paper. And if you have any questions about the paper, always reach out to me, and I'm happy to kind of describe. It's very scientific, as you can imagine, and I know you're not all scientists, so happy to try to explain it to the community at any point. But I guess that summarizes it. Our lab's doing well, and I think that we've made some nice progress in the field, as Denise has said, and we continue to be very dedicated to continuing with that progress, and we will continue until we have really, really good therapies for EHE, and we're bored of studying it, but I can't imagine that that's ever going to happen. All right.
Thanks, Dr. Rubin. Thanks so much. Great, great overview. We appreciate the update. Thanks for giving us time between all the sporting events today too. Enjoy basketball. So I want to go next and talk a little bit about MSK. So many of you might be familiar with the Memorial Sloan Kettering Cancer Center International Center of Expertise in EHE. This is a collaboration that was formed officially in 2019, but truly well before, and Dr. Rubin just did a shout out to Dr. Antonescu. Well before 2019, Drs. Tapp and Dr. Antonescu were studying EHE, and Dr. Antonescu very early on publishing about EHE. But this collaboration really was formed in order to build a foundation of clinical and science understanding about how to tackle EHE. So Dr. Tapp, along with his team of medical oncologists, committed to forming a multidisciplinary team to treat EHE. Through that, obviously, patients get to come into that institution. They can consent to be participants in research that is occurring at that institution. Things such as molecular screening, tumor banking. So if they're having a procedure at MSK, then those tissues can be banked in MSK's institution for further studies. Dr. Antonescu and her lab have been, again, for a very long time, studying EHE and looking at the tumor microenvironment, immune filtrates, and they still have work that they're doing in order to build a different type of EHE model called CRISPR-Cas9. I'm not going to even try to describe that. Dr. Rosenbaum has been with Dr. Antonescu, have published on clinical and molecular subsets of EHE. And again, we think that MSK may have the largest by numbers cases of EHE in the world. We don't know that. There's no solid data on that, but we know that there are a lot of cases at MSK. They have a very large medical oncology team complemented with Dr. Antonescu's lab and others to study EHE. So really, really groundbreaking work going on there.
Okay, this is for me. This is the largest international collaboration in EHE research that we're funding at the moment. It's a joint project between Istituto Nazionale dei Tumori, or INT in Italy, and the Royal Marsden Hospital and the Institute of Cancer Research in London. And actually, although we think of it as one research program, it's actually 3 different projects that we funded, and I've color-coded them here. The first project was to try and identify a biomarker or biomarkers for EHE. Biomarkers are substances in our blood that are related and correlate to the EHE. And so if those biomarkers go up, it maybe suggests that your disease is becoming more progressive. It could also be used for diagnostics. And it could also, if they correlate directly to the aggressiveness of the EHE, it may give us early signals as to whether, for example, a drug therapy is working or not. And the importance of that is not only because it helps with prognosis and diagnosis, but if it allows us to identify these movements in the aggressiveness of the disease earlier, then like all cancers, the earlier we identify what's going on, the better our chance of trying to deal with it. So that's what they've been doing. And in fact, they have found a product called GDF15, Growth and Differentiation Factor 15. GDF15, which in a patient cohort of 13, they identified as potentially being a biomarker. And they've now verified that in a second cohort of patients of 15. So that's great news. So that looks like the first biomarker is beginning to develop. Hormones, we haven't made much progress. And with the microRNAs, they've changed tack here, because rather than trying to identify individual microRNAs and then work out if they're related to EHE, they're going at it in a different way, in that they're going to use RNA sequencing to identify all the microRNAs in a patient's blood and then in healthy people's blood, and actually look for the differences and see if they can identify that way microRNAs that are connected and clearly in patients with EHE and not in people without EHE. And microRNAs are just tiny fragments of an RNA that are non-coding. But they are involved in the disease progression in other ways, so important to find out. The second project in black was starting with trying to develop 4 more PDX models. INT have a PDX model. That's what, as Denise explained, that's one where we've taken human tissue and put it into the mouse. So the mouse is acting like a life support system. The tissue grows, and as it grows, they take bits out and they put it in more mice. And so they end up with more and more mice with human tissue in that's growing. And from those mice, they've been able to create a cell line, which they are also being able to use now in some of their research. And ultimately, with those models, and the idea of why 4? If you've got one, why do you need more? I think Dr. Rubin always says you can't have enough models. And the point is that they're trying to create models of different forms of EHE from different patients. Now, I think they've achieved 1 more. So far, so that's good progress. We hope more are going to come, and the whole point of these models is to then use them in different ways to ultimately drive us towards new therapies and new drugs that can help treat this disease. They're already looking at combinations of drugs and new drugs on their own. One of the focuses, of course, is sirolimus, trying to understand how that works and how we could improve it. They're going to use genome sequencing to try and identify genes that are either resistant to drugs or sensitive to drugs. And that may allow us to then identify positive therapeutic possibilities. And as we talked about, they're going to do INT particularly has a real expertise with these microRNAs, and they're hoping to use those to help them try and understand how EHE tissue is responding to different drugs. So lots of work going on there, as I say, driving towards new treatments. And then the third, and in red, is the observational study that we're about to start. This is quite exciting. We're hoping to have about 20 hospitals across Europe involved in this, collecting prospectively data on EHE patients over the next 3 to 5 years, which will hopefully provide us with a new and pretty thorough database about the disease and what's happening. So a great project overall, going well.
Okay, let's talk a little bit about Dr. John Lamar's lab at Albany Medical Center. Some of you might have heard Dr. Lamar has spoken at a couple of our conferences in the past. And this is an excellent model for collaboration. So very much like the Rubin lab, and the Rubin lab and the Lamar lab collaborate regularly. In fact, the cell lines that have been developed in the Rubin lab have been parlayed over to the Lamar lab and are enabling that research. And there's new work drives more work and different work. So they're making some adjustments in their research and advancements because of the cell line that's been developed in the Rubin lab. So there are 3 we've broken this down in Dr. Lamar's lab, and I know that there's more behind this, but there are 3 keys. Dr. Lamar's lab focuses on molecular pathways to target or inhibit the function of the fusion proteins. And through various fundings, the EHE group have funded the Lamar lab, and through other private donations, his work is funded to identify different pathways that can repress the function of TASCAMP1. and then test to see if targeting those pathways can inhibit the growth of EHE. So there's a lot bundled up into that. He has a very recently initiated study in the middle of 2022 that is going to identify those druggable pathways to treat EHE. And the idea is to look and find FDA-approved drugs, this is drug repurposing, that'll block that TASCAMT1 function. And then once those drug candidates have been identified, then use those cell line models, to look at repurposing in patients. These are projects that actually could translate into products that are treating EHE, people living with EHE today. So a talented group of people at the Lamar lab. John Lamar is, he's a professor. He's breeding new young scientists. He's growing them in his lab. He is out there. He and Guy Weinberg work on the co-leaders of the Telluride Science Summit. So just a lot of good EHE focus coming out of this lab. And then switching gears to our Canadian partners. So Dr. Rozek, if you're not familiar with Dr. Rozek at Princess Margaret Cancer Center, ProCare is possibly the newest, most significant, we'll say EHE initiative started in the world, I think. This initiated very, very late in 2022 officially. I think it's been in the works for quite some time. But again, led by Dr. Rozek at Princess Margaret, but this is going to be a research program that is going to happen across Canada. And ideally, it'll be a multi-center initiative, and it'll harness and curate data, building a virtual tumor bank, EHE tumor bank, and that virtual tumor bank will be used to help create more EHE PDX models, which will also be used to evaluate. They're going to look at evaluating mTOR inhibitors. They'll have a patient gathering, data gathering component to this. So gathering information on patient treatment utilizations. I think patient perspectives are going to be involved. So this is a very, very significant initiative that's going to start happening. If you are joining from Canada, please, make sure that this is on your radar. Take a look if you are, if your clinician is talking with you about this, please, if you're able to join a study or an initiative like this, we would encourage you to do so.
Okay, this is another great example of an international collaboration that has been going on for some time now, and this involves Australia, the US, and Canada. And the team here in combination have been focusing on trying to understand how the fusion proteins TASCAMT1 and YAP-TF3 are affecting the Hippo pathway and driving the formation of tumors. And you'll remember I mentioned the Hippo pathway a little bit earlier on. It's the process that regulates cell regeneration and growth. They are not only just looking at the fusion proteins, but they're also trying to understand other products or compounds in our cells that may be impacting the overall transcriptional process. And you remember transcription is at the start of protein production in our cells. And I think actually published a paper, I think it was this team, apologies, I don't absolutely know who published, they think they identified the third protein that connects on the other end of TASCAMT1. When it connects to TEAD, they'd identified the third protein. I think I may have got that wrong, but we'll see. So anyway, this team is continuing, like others, to try and get into the real detail of the biology of EHE, understand it, and as they do that, hopefully again identify new therapeutic targets that maybe we can use to either stabilize EHE or actually find a cure. And I think it's really exciting. All of these research projects have this underlying theme of finding new therapeutic targets. This is a project that was undertaken in the University of Manchester. It's a 4-year PhD that has just finished. And in this project, the team developed a cell line using embryonic stem cells, pluripotent stem cells, mice cells, which are then differentiated to endothelial cells. And TASCAMTA1 expression is induced with doxycycline. And they were able to then look very, very quickly at some of the effects of TASCAMTA1 in the cells. One of the first very early things they did, for example, is they looked at all the genes that are differentially expressed in cells as a result, or what appears to be as a result of TASCAMTA1 expression. And that was a very large number. Over 4,500 genes were differentially expressed. But what was good is that in those genes that differentially expressed, they saw the correct differential expression of the EHE gene set. So we think this is a good model for EHE. What they went on to look at was what happens in the cell cycle with EHE cells. The cell cycle is the circular thing you see here in the top right-hand corner, and it's the cycle that cells proliferate round and are regenerated as they go round. And what they found was that EHE cells were becoming trapped in the S phase of the cell cycle, which is at the bottom of that diagram. And that's the phase where DNA is replicated as the cell divides and creates a new cell or multiple cells. And so they started to look at the DNA damage as an issue. They showed that there was double strand break DNA damage, which was causing the arrest in the cell cycle. And as they went on, they began to develop a possible model that suggested that cells that were trapped in the S phase either then became senescent, which means dormant effectively. They don't die, but they just do nothing. They just sit there. And that could be what we see when we see indolent disease. But what they also found was that the main repair pathway for DNA damage, homologous recombination as it's called, was also impaired in TC. And what happens in that case is that other less competent repair pathways kick in and start trying to repair the DNA damage. A bit like a mechanic, you know, the mechanic who knows your car isn't available, so another mechanic comes along who doesn't know your car and creates complete chaos. And so what happens is that actually, rather than repairing the damage in some cells, it creates secondary gene mutations. And I think Dr. Rubin talked a minute ago about the implication of some secondary gene mutations may make the disease more aggressive. And in this model, the theory is that in some cases, those secondary mutations actually allow the cell to bypass the arresting process in the S phase and continue round the cycle. And so now you have progressive disease. What's interesting here is that the EHE Foundation have just funded the team in Manchester in the next 6 months to look at PARP inhibitors. And these are drugs that have been used for other cancers where genetic mutations are an issue. And it's sort of, I always think it's slightly counterintuitive because the PARP inhibitors stop the DNA repair. And so what they do is actually force the cells to remain damaged. And in some cases, the body recognizes that damage and kills the cells. That's part of your system. The body is usually very good at recognizing cells that are damaged and just killing them off. Okay, finally, very quickly, just a general overview of or comments about Australia. Australia, we are funding some work down in Australia looking at the genetics. But what I wanted to talk about with Australia was to talk about their research infrastructure, because they have developed what I think is an excellent set of studies and registries and biobanks, etc., that all sit under umbrella organizations and which can be integrated, accessed by researchers. They can access multiple registries, etc. And it's actually a very powerful overall situation that they developed. I'm not going to go all through these. Denise mentioned ISCIS. ISCIS is where they're taking genetic samples from sarcoma patients to make available to researchers Our foundation in Australia is funding the genomic analysis of the EHE patients to see if there's anything that comes out of that that could be of interest. And I think we have something like 70 EHE patients who've donated samples to the ISCIS project. So a very good setup. And the WEHI at the bottom there, the WEHI Rare Cancer Research Program is a very, very good integrated rare cancer research program. And they are running or helping with the collection, for example, of EHE samples. And we have been able to put them in touch with the INT team in Milan, who have developed a PDX because they're struggling to develop a PDX at WEHI. And so that's a great example of where we as a patient community can actually put people in touch with each other and try and help them, because if we can help someone create more models, that's fantastic. Okay, over to you, Denise.
Thank you. Okay, so I'm aware that we're running a little bit slower on time. We only have a couple more slides to go through, and then we'll get into Q&A. So just want to let everybody know this is not going to go on forever and ever. But let's talk quickly about drug repurposing, which is a faster way to find treatment. So drug repurposing essentially looks for drugs that have already been proven to be safe in humans, and then figure out if they're going to work in EHE. So we need to look for drugs or drug classes that might work better or best. And there are 3 projects over on the left side of the screen that we funded so that labs can look at that. So Dr. Pabati at the Cleveland Clinic has a project that's going on this year to look at FDA drugs they're already screening. And then John Lamar, we talked about that. He already has a project ongoing. That is a multi-year project. And it's a different way. So each of these projects are different in how they approach drug repurposing and screening for these drugs. And then we've talked about Dr. Kouskoff's model and looking at PARP inhibitors. So once that lab work, that preclinical work is done and some data is generated, then essentially you need that data to support regulatory bodies to give you the approval to look further at that, because you need regulatory approval to do the studies. And then you generate more data, because we need drugs that are approved for EHE. Off-label, we'll say, limits access to drug. So we need label approval for EHE. So in the US and EU, there are very, very large and focused projects to repurpose sirolimus. Many of you are familiar with, and some of you may have participated in the Mechanism study. Dr. Schutze headed up out of the University of Michigan. That trial has results. It did not meet its primary objectives, and I think you'll see final data coming out of that. I would expect sometime mid this year or later this year. But these are the types of things that we need to do, because again, we can go through the long clinical trial process, but if we can use drugs that have already been proven safe in humans, that will get us there a little bit faster. And then I want to talk just quickly, and we talked about this, Dr. Rubin's at the Cleveland Clinic, Dr. Dale Shepard actually is the investigator on the Novartis phase 1 clinical trial of a TEAD inhibitor. ICANA has a phase 1 clinical trial of a TEAD inhibitor. You've seen both of these in our collective news, and we've listed here a couple of very relevant immunotherapy studies. On the right, you see just a little image that'll show you a little bit about the clinical development plan and how these phase 1 studies, this is the ICANA study that's shown here. This is public information, so we're not sharing anything that's proprietary. But essentially, as Dr. Rubin said, you're looking at toxicity. You're looking to see if a drug is safe in humans, and they escalate the dosages of that drug to figure out what the safe dose is, and then start adding dose expansion cohorts. So you'll see that EHE is in a cohort in the dose expansion phase of the ICANA phase 1 study. So I think that the big takeaway on clinical trials is the list of clinical trials for EHE is growing larger. The locations The availability of clinical trials to EHE patients specifically is global right now. You can see there's something everywhere. And we expect this to continue to grow through 2023. So definitely stay alert to that. If you're looking for a clinical trials are an excellent part of care for cancer patients particularly, but in general, I'm a big advocate for clinical trials as a part of your care considerations. It may not be right for you, but talk to your doctor. You have to be your own advocate. You have to go looking. Your doctor may or may not be conducting a trial, so they may or may not bring you those opportunities. So definitely stay alert to news on those. And, you know, summarize what we've tried to impress here and will continue to impress as we look forward and bring you more projects, more opportunities to participate in research. EHE patients are making a difference. EHE patients are the only people who can give their data for research. Your doctor can give data on your behalf, but you can give data directly to research, and it's so important. Biobanking, we just simply need it. We need a lot of it. 5 and 10 samples will do a little bit, but Dr. Rubin also talked about multiple attempts to make cell lines. These don't always work out, and you need continuing samples in order to keep on making models. And collectively, if we can keep building data and information and building bigger databases that can be harmonized across the globe, and we can continue to get biological samples, fundraising is key. None of this happens without funding, and we can't rely on the government and we can't rely on pharma to always bring in that funding. We're getting some heads turned in that direction, but we need fundraising. And all of that together gives us momentum, and we're going to find a cure. We're going to find a cure, or we're going to find better treatments for this disease. Hugh, you want to wrap us up?
Yeah, so that's really the end of the description. We just wanted to finish by saying a massive thank you to all our patient community who have already provided data and samples and information, and all the supporters, not just patients, but their supporters who've provided unbelievable levels of fundraising and have enabled a lot of this work, all of this work to go ahead. But it's what I call a great start, but there is much more to do. And so we can't sit back and say, oh, great, well, we've done that now. And the wonderful thing about our research is that actually all of these models all of this research is producing more and more research for us to look at. And that is a wonderful problem to have. It would be terrible if we were sitting here saying we've done all these projects and we haven't found a single thing of interest. That would be utterly depressing. It's completely the opposite. We need to keep going, and we need to drive this on, and we need to give. We've got some fantastic people in our corner. Dr. Rubin, Dr. Lamar, Dr. Stacchiotti, Robin, all of these people, they are there fighting the corner for all our patients, and we've got to be there with them, producing data and information. So when we ask, please join in all of these initiatives, because it really is so important. Okay, enough from us. Q&A. Let's go.
I'm going to jump in here for a quick second, because for those of you that have to jump off now and aren't going to be able to make it to the Q&A, we are now going to go into the Q&A. But first, I wanted to thank Denise and Hugh and to everybody attending from around the world today. We hope that this Community Connections has been helpful to you in some way. I want to give some reminders that our next Community Connections will be Saturday, February 11th, 2023 at 2 p.m. Eastern Time. It's our fourth session with Dr. Vessel, Chief of Division of Palliative Care, Clinical Associate Professor of Medicine and Pediatrics from Tufts University, and Dr. Mae Beckler, Life Cancer Coaching, and they will continue their discussion on who I am now, integrating the cancer identity into the self. They gently guide insightful discussion on personal feelings surrounding diagnosis and living with EHE, and it has been helpful to so many. If you guys have any ideas for future sessions or questions, you can always email us at info@fightehe.org. And a few reminders, the things that are coming up to mark your calendars. April is EHE Awareness Month, and in April we will be having our annual virtual fun run and walk, and we will also be holding our EHE 360 Global Patient Conference on April 15th. So for those of you that are signing off now, thank you so much for joining us. We're sending you love and hugs and just live. For any of you that can stay around, if you have questions for Hugh and Denise, we will start that Q&A now. Go ahead, Denise.